So good afternoon, everybody, and welcome to this ERKNet webinar. My name is Francesco Emma, and I'm speaking from my office in Rome. As usual, before starting, let me remind you that your microphones are turned off, but that during and after the webinar, you can send your question through the GoToWebinar application that appears on your screen. I will ask your questions at the end of the presentation. The webinar will last approximately 30 minutes, and then we will have 15 minutes available for your questions. Our speaker today is Leonardo Salviati. Leonardo is a professor of pediatrics at the University of Padova in Italy, where he is the current director of the residency program in medical genetics. Dr. Salviati research has focused on the molecular basis of neurological and metabolic diseases, in particular mitochondrial disorders and disorders of the intermediate metabolism. He has co-authored more than 130 articles in peer-reviewed journals, including some seminal articles on genetic mitochondrial disorders causing also renal diseases. Today, the title of his lecture is Steroid-Resistant Nephrotic Syndrome in Mitochondrial Cytopathies. Please, Leonardo.
Thank you, Francesco. It is a pleasure for me to be here today. And but let's get directly to the topic because we have a lot of things to say and we don't have much time. Okay, so I think you all know what mitochondria are. And the mitochondria are these small intracellular organelles that originated from ancient bacteria similar to Rickettsia that enter in symbiosis with the progenitor of the eukaryotic cell. Now, the structure of mitochondria resembles that of gram-negative bacteria, so they are lined by a double membrane, and the inner membrane has these protrusions towards the inside of the mitochondria called cristae. The cristae are important because their morphology is critical for mitochondrial function, and cristae host the enzymes that of the mitochondrial respiratory chain. The mitochondrial respiratory chain is comprised of 5 complexes from name complex 1, 2, 3, and 4, and 5. And the first 4 complexes make up the electron transport chain. So what they do is they take electrons derived from the oxidation of carbohydrates and lipids mostly, and through the Krebs cycles, and they transfer these electrons to oxygen forming water. This produces energy which is used to pump protons outside of the mitochondrial matrix into the intermembrane space. Now these protons then enter through complex 5 or TPase, which exactly like a turbine engine, they allow to produce ATP. Now, so this is basically what electrons in mitochondria do. These are they either from complex 1 or from complex 2, but also from other enzymes in the mitochondrial in mitochondria, for example, beta oxidation enzymes or others. And all these electrons generated by these reactions are transferred to coenzyme Q. We'll see that this molecule is crucial in a number of disorders of nephrological interest. And these electrons ultimately, as we said, are used to make transfer to oxygen and to make water. Now, why do we need so many complexes? Because this allows the energy transfer to be gradual. Basically, the reaction that takes place is the simple combination of hydrogen and oxygen to produce water and energy. But only if the reactions occur in a gradual way and controlled way, a great amount of energy can be actually used in a purposeful way. So it can be transformed into chemical energy. If this happens in an uncontrolled way, well, this is what happens. The same reaction occurring in a violent, not uncontrolled way. Just consider that the thermodynamic efficiency of mitochondria is almost 40%, which is incredible if you think of what are the best internal combustion engines that are available today. However, mitochondria are not only required for ATP production, but they also perform many other functions that are equally important. For example, they are involved in the control of apoptosis, and they are also essential for calcium homeostasis and for a number of other reactions. For example, pyrimidine biosynthesis is of course also in mitochondria, beta oxidation, urocycle, and many other reactions of the intermittent metabolism take place within these organelles. Now, the interest in mitochondria derives from the fact that mitochondrial dysfunction causes common human diseases. There's an estimate that calculate that their combined frequency could be up to 1 in 5,000. So it's they are frequent. They're rare. They're frequent rare disease. And however, we must make a point now. Throughout the years, there's been a debate on what should be considered mitochondrial disorders. And there were two interpretations. A wider interpretation, that is any enzymatic or functional defect affected by the mitochondrial, while a more restricted interpretation, which is the one that we will follow today, focuses on defects of the respiratory chain. Now, these defects can be direct or directly derived from the specific mutations we'll see, or could be also indirect. Anyway, this is what the mitochondrial disorders are due to dysfunctional respiratory chain. Now, the peculiarity of mitochondria compared to other organelles is the fact that they have their own genome. In fact, if we look at the human genome, we can see the 22 autosomes, chromosome X, chromosome Y, but also the mitochondrial genome. Now, the mitochondrial genetics do not follow the rules of Mendelian genetics, but it's a system that is more. These laws are more similar to those that we find in population genetics. So when we deal with mitochondrial genetics, we see we have a completely different set of laws that regulate the disease transmission and also the appearance of the disease. Now, mitochondrial DNA is similar to bacterial chromosomes. In fact, it's what remains of the genome of the ancient bacteria that enter the symbiosis with the progenitor of the eukaryotic cell. So it's circular. It does not have introns, so it's almost all coding, and it's not organized into chromatin. Although there are some mitochondrial DNA binding proteins, you never see an organization as complex as you see in nuclear chromosomes. Now, and also there's also another point is that the mitochondria actually have a different genetic code. So that implies that they cannot use the mitochondrial, the classical cellular protein synthesis machinery, but they have one of their own because they have their own genetic code. Now, mitochondrial DNA encodes for only 13 proteins, but also for the 22 transfer RNAs and the 2 ribosomal RNAs that are required for protein synthesis. These 13 proteins are all components of the mitochondrial respiratory chain. So this is the mitochondrial DNA code. You see these underlined, these are the codons that are different from the universal genetic code. And these are the most important rules of mitochondrial genetics. So maternal inheritance, you know, that means that the mitochondrial DNA is only transmitted from the mother, but mitochondrial DNA defects can affect both males and females. Heteroplasmy, threshold effect, and random drift. We will see detailed what these other terms mean. Now, in classical genetics, you have two alleles for each gene. So basically, this allows three situations. Either a person is healthy, or has a heterozygous mutation, or has a homozygous mutation, meaning that both alleles are affected. So you can have 0, 1, or 2 affected alleles. There's nothing in between. So it's like quantum physics. You have 3 states. That's it. While mitochondrial genetics is more similar to classical physics, in that you have a continuum spectrum of situations. So when you have a mutation, it may affect different proportion of mitochondrial DNA molecules, because in a single cell you find there are many mitochondria, and each mitochondria contains many genomes. So basically, you can have 1 in a 1,000 mutated molecules, 2, 3, 4, and that's it. So you have a continuous spectrum of situations, not something as defined as in classical genetics. Now, you must also, however, consider that for the biogenesis of the mitochondrial respiratory chain, you need both nuclear DNA and mitochondrial DNA. And we've seen that mitochondrial DNA encodes for these 12 protein subunits. All the others, more than 60 subunits of complex 1, 2, 3, and 4, and 5, are encoded by the nucleus and imported into mitochondria. But the nucleus also is essential for encoding other proteins required for the biogenesis of the respiratory chain. For example, all the proteins that are required in replication and maintenance of the mitochondrial DNA, they're all encoded by the nucleus, as well as RNA transcription or protein synthesis. Coenzyme Q biosynthesis enzymes are encoded by the nucleus, and then these proteins are transferred into mitochondria, where coenzyme Q biosynthesis takes place. So you have many hundreds of genes that are required for the biogenesis of the respiratory chain, encoded by the nucleus. So basically, you have these two systems, mitochondrial DNA and nuclear DNA, and their cooperation is what allows the biogenesis and the correct functioning of the respiratory chain. Now, we can envision a sort of a classification, of genetic classification of these disorders. We can have nuclear DNA defects, and these are basically follow the standard rules of Mendel. Or we can have mitochondrial DNA defects, and they follow the rules of mitochondrial genetics. So there's heteroplasmy, the threshold effect, and all the other stuff. But we also have a third category, in which, and these are mitochondrial DNA defects that are not primitive, but they are secondary to a nuclear DNA mutation. And most of these are caused by mutations in nuclear genes that are required for the maintenance of mitochondrial DNA. The most common manifestation associated with respiratory chain dysfunction is tubulopathy. And this is for a simple reason. Tubules, the tubule, especially the proximal tubule, is rich in mitochondria because its main function is to perform reabsorption of molecules from the lumen of the tubules against a gradient, and so they require a lot of energy. On the other hand, the glomerulus has few mitochondria, and in fact, glomerulopathy is rarely seen in mitochondrial disorders, except for a specific set of defects, which are defects of coenzyme Q biosynthesis. There are also some specific mitochondrial DNA mutation that may be associated with mitochondrial dysfunction, with glomerular dysfunction. Now, there's a recent paper that came out last year in Cell Reports by Tobias Huber's group, in which they provide a nice representation of the fact that the glomerular cells rely mostly on glycolysis, and in fact, if you disrupt genes that are important for mitochondrial biogenesis, and in terms of energy production, they do not usually cause glomerulopathy. So that means that the mechanism of glomerulopathy is probably different than what is causing tubular dysfunction and in general of other mitochondrial disorders. So it is not due, the glomerulopathy is probably not due to deficient energy production, but to something else. Now, there are many more, several hundreds known mutations on mitochondrial DNA, but the most frequent is this mutation affecting the nucleotide 3243. This is within the gene for a tRNA, so it's not for a protein coding gene, but it encodes one of the tRNAs that are required for protein synthesis. Now, these are two families that we studied in past years, and in which you can see there's a sort of the mother was affected as well as her two sons, and they had a combination of hearing loss and proteinuria, and one of the two also had diabetes with onset in adolescence. And they had they harbored, all three harbored this specific mitochondrial DNA mutation. And another family we had is even more curious, because this is a three-generation family, in which taking the family history, we learned that the grandmother of the index case was this boy here, had renal problems and had died of cancer at around 50 years of age. And the boy, the index case, had diabetes, hearing loss, sorry for the typo, encephalomyopathy, and renal failure. But also his mother had proteinuria and hearing loss. We could not study the mother's sister, his aunt, but her cousin had apparently isolated renal failure. Now, what is interesting, when we studied this boy, we found that it was the clinical picture was suggestive. We didn't even sequence the entire mitochondrial DNA at first, but we went directly for the Milas mutation. In fact, we found the Milas mutation. What is interesting is that we found the mutation also in the other members of the family that we could study. And you can see a correlation between the levels of the mutation. You see these numbers here are the percentage of mutant DNA in various tissues. Here is RB stands for renal biopsy, US urinary sediment, BS is buccal smear, HF is hair follicles, and BL is blood leukocytes. And you see that, for example, there's a correlation. This boy had had high levels of mutation also in many other tissues, while his cousin that had isolated renal manifestations had low levels of the mutation in the other tissue that we could study. So there is a correlation between the levels of heteroplasmy and the manifestation. This is not always the case. What is curious in this family is that the mutation segregated preferentially in all members in the kidney, and this is not the case. Actually, it's only a minority of patients of MELAS patients who actually develop renal symptoms. In all, we have 3 families, and that's it. The majority of our MELAS families actually do not develop renal failure. Why is this only for the MELAS mutation? We don't know. Other tRNA mutation usually do not cause renal glomerular problems. They mostly cause, as we said, either tubulopathy or tubular interstitial nephritis, but not glomerulopathy. We have also we thought that this had to do with specific functions of the tRNA that were not directly linked to energy energy production or respiratory chain biosynthesis. But now we found another patient we still have to publish. She had a complex multisystem picture with proteinuria and microhematuria since her early teens, and then she developed cataracts and hearing loss. And so it, of course, was suspected Alport syndrome, but the EM was not suggestive of such diagnosis. And given this clinical picture, when we saw her in the clinic, we thought about a possible mitochondrial DNA mutation. We sequenced the whole genome and we found a mutation in cytochrome B. Now, cytochrome B encodes for a component of complex 3. It's and this is a pathogenic mutation. It's a truncated mutation, so it completely abolishes cytochrome B function. You can see there is it's heteroplasmic. You can see both the wild type and the mutant alleles in this PCR of FLP. And what is important is that it's enriched in urinary sediment. Unfortunately, we could not study the kidney, but we guess we could have found high levels in the kidney. Now, I'm showing this just to point out that it's not clear why this protein coding gene that actually was associated with glomerular rather than tubular problems. We don't have a clear explanation of this. The other important things I want to tell you is that even if you perform standard whole exome sequencing, you will not find these mitochondrial mutations. You must study the mitochondrial genome. This is for a number of reasons, and if you study the mitochondrial genome, the best place to do it, if you can, is from urinary sediment cells, because if you don't have muscle available, because for again still unclear reasons, pathogenic mutations are enriched in uridine binding segment cells. Now, the last part of my talk will be devoted to coenzyme Q10. Now, coenzyme Q10 we've seen is a component of the respiratory chain, but it's not a protein; it's a lipid. It's a redox active lipid that is formed by a long lipophilic tail and a polar quinone group in the head, and this is what binds electrons. Now, coenzyme Q, as we've seen, is involved in multiple cellular function. That is, the most important is the production of energy in the respiratory chain, but also it's one of the main cellular antioxidants. But it's also involved in control of apoptosis because it modulates the permeability transition pore. And it was recently found to be important also for detoxification of hydrogen sulfide. The biosynthesis of coenzyme Q is still relatively not completely known. We know that two components are derived from acetyl-CoA, the tail, and the quinone group is derived from tyrosine. This set of reaction is still only partially known. And then the final set of reaction, which takes place in mitochondria, modifies the quinone head. So this is what we until this year, this was the commonly accepted model of coenzyme Q biosynthesis. You see that two enzymes are still missing. We don't know what these are. And there are many genes that are thought to be involved in the process. Just to make things difficult, they're called CoQ1, CoQ2, CoQ3, CoQ4, CoQ5, and that's it. And this is the nomenclature derives from yeast. And these genes, then there are some genes who are really do not, there are really whose function is not completely known. Now it's, for example, these two genes that now are called CoQ8A and CoQ8B are regulatory proteins. What is important that all these proteins are gathered up in a multi-enzyme complex, which carries out this reaction. And coenzyme Q deficiency, it can be associated with many different phenotypes, affecting the majority of tissues of the organs in the body, the brain, so encephalopathy, different types of encephalopathy. The kidney, and we'll see that steroid-resistant nephrotic syndrome is a hallmark of coenzyme Q deficiency. Rarely can also give tubulopathy. The muscle and also the heart and the liver and many others. Now, there are many genetic defects known till now affecting all these genes, and we can divide them in two great categories. Those that cause nephrotic syndrome, and those that don't. This is just a very simple classification based on the phenotype. And we still don't have an explanation why some of these genes cause steroid-resistant nephrotic syndrome and some do not. They cause only encephalomyopathy. We also now know that the set of reaction is different than what is previously thought, and that and this is what is now the most recent model of how the biosynthesis takes place. And you can see that in red are the genes that cause nephrotic syndrome. in blue the genes that do not cause the nephrotic syndrome, and you see that there is not a real division in the real, I mean, explanation of why some do and some don't, because so these are all enzymes, and they cause nephrotic syndrome. This is also an enzyme, but it doesn't cause nephrotic syndrome. So it's not that the regulatory subunits are associated with nephrotic syndrome and yet the non-regulatory ones, the enzymatic, do. It's simply we don't have an explanation. Now, the why is important? Because to recognize coenzyme Q deficiency, because this is what we published back in 2005 with Giovanni Montini. We saw this patient who had nephrotic syndrome, a steroid-resistant nephrotic syndrome, and then developed an encephalomyopathy. And we administered coenzyme Q10 orally to this patient. We gave high-dose Q10, and we could stop the progression of the encephalopathy. Now, what is important, when we still didn't know the gene at that time causing the disease, but when we saw his sister, who also developed nephrotic syndrome, we treated her immediately, and the proteinuria was stopped. Now we have a much longer follow-up, and now it's all more than 50. You have more than 15 years follow-up, and we have to say that the renal condition gradually worsened, and she was transplanted this year, but she entered dialysis only relatively few months ago. So we could still buy her time, and while the brother had in few months he progressed to renal failure. And we know that also coenzyme CoQ6 patients and CoQ8b ADCK4 patients respond to coenzyme Q supplementation, and also the mouse model. Also in the mouse model, you can see progression of disease despite treatment. So it's not that the treatment was done too late in this case, but it's the disease that is you can observe a response, but it's not completely controlled in the long term by coenzyme Q. And this is really nice. You see that even if these are the untreated, the wild type is treated with idebenone, which is an analog of CoQ that I warn you not to use, because you can see here that it doesn't work. You can see here that the mice treated with CoQ, the mutant mice, and you can still see that there is some progression of the disease. It's not 100% survival, but still it's much better than the untreated. Now, so if it's not with the bioenergetic defect, what is causing the renal failure? Well, we don't really know. There are many things that go on when you take out CoQ10 from cells. You do have the respiratory chain deficiency. You have an increase in oxidative stress, and you have an impairment of pyrimidine biosynthesis, an inhibition of beta oxidation, and reduced activity and clearance of sulfides. You also have an increase in mitophagy, which is in this case is protective, because if you block it, in cellular models, these cells die. Now we can skip this. The group of Micho Irano has made an interesting observation some years ago, is that there is a weird relationship between coenzyme Q level, residual coenzyme Q levels, and ATP production and ROS production. And that is that at very, very low levels of residual coenzyme Q, so you never have zero, because zero is incompatible with life in patients, and also it's incompatible with life in cells. So you always have some residual coenzyme Q in cells. But for very low levels of coenzyme Q, you don't have ROS. Why do you don't have ROS? Because the respiratory chain is blocked. So if it's completely blocked, you don't make ROS. And you have severe ATP deficiency. While if when you increase residual coenzyme Q because you have milder mutation, the ATP production gradually normalizes, but you have an increase in ROS production, which begins to drop only after you have more than 50% residual CoQ. So at low levels of CoQ, the ATP deficiency predominates, while at higher residual levels of CoQ, you have less marked ATP deficiency, and while you have mostly ROS, an increased ROS production. And this is in fact the PDSS2 mice, which have this mutation in CoQ1, have widespread CoQ deficiency, but display increased ROS only in the kidney. And another important observation was that if you knock out this gene in mice, only when the knockout occurs in podocytes, but not in other renal cell types. So it is specific for the podocytes. And so this is what we think is happening. As we were saying here, at very low coenzyme Q levels, you have low ATP but also low ROS. So the mitochondrial symptoms prevail. So you have the clinical pictures that are very similar to other mitochondrial disorders. You may have tubulopathy in some cases. In intermediate CoQ deficiencies, you have increased production of ROS. So you have a combination of classical mitochondrial symptoms and nephrotic syndrome. And with milder CoQ deficiency, you have isolated nephrotic syndrome. And no other mitochondrial symptoms because you have enough ATP. This is shown well by CoQ2 defects, and the CoQ2 is associated with a variety of phenotypes that range from fatal multi-organ failure at birth to isolated nephrotic syndrome in the second decade. And we have shown that there is a nice relationship between residual function, residual CoQ production, and the phenotype. And basically, when you have severe CoQ deficiency, you have these phenotypes. When you have milder deficiency, you have the nephrotic syndrome, and which can be even isolated or, in the more severe cases, accompanied by neurological symptoms. And this phenotype correlation is clear for CoQ2, and also for CoQ PDSS2. We are working on that. But instead, for CoQ8B, which is another protein, and another gene associated with the steroid-resistant nephrotic syndrome, this is not true. And we didn't see much difference between patients with homozygous truncating alleles and homozygous missense alleles with residual CoQ production. What is interesting is that we found a common polymorphism in CoQ8B, this arginine in position 174, that there are two alleles, arginine and histidine, which are almost equally present in the population. Well, it turns out the histidine allele is less stable. So we hypothesized having the histidine allele could be a risk factor for or a modulation factor for other defects. And in fact, a paper just came out in which this hypothesis was confirmed, and we have a new CoQ6 family in which we also observed that the presence of the histidine polymorphism was associated with a more severe clinical course, having the two patients had the same CoQ6 mutation. This is now last 5 minutes. a few words on also this nice paper that came out last 2 years ago by Caterina Quincy, showing a possible role of hydrogen sulfide in causing the nephrotic syndrome. However, this paper needs to be confirmed. And this other thing I wanted to show you is just what is being tried now for the therapy, at least of some of these forms, and this bypass therapy. And this is an observation made by our friend Fabien Pierrel, and who noticed that if this is the precursor of quinoline of coenzyme Q, and this is the CoQ6 is the protein is the enzyme that catalyzes the hydroxylation of this carbon number 5 in this position. So CoQ6 puts an oxygen in this position, then CoQ3 puts the carbon here. So he hypothesized if you give this molecule here to the cells as a precursor, you could bypass the defect. And in fact, this is seen in this slide. We tried to do it on the human mutants using, this is a yeast model, and you can just see that in these cases, where there are the yeast that express these mutations do not grow. But if you add these two substances, in particular VA, to the medium, the yeast start growing again. So you can sort of bypass the condition. And this was the same experiments done on human cells that lack CoQ6. And you can see that VA completely restores CoQ10 levels and ATP production and respiration. And what is more interesting is that this VA compound also normalizes ROS production, which is much higher in the cells that receive CoQ compared to the cells that compared to the wild type cells and the cells treated with VA. So this could be an explanation of why, in the end, with CoQ supplementation, you still have the nephrotic syndrome, because the kidney is subjected to chronic ROS damage. And what is VA? Is vanillic acid. So a common food supplement that is used in pastry and so on. And it's cheap and absolutely non-toxic. And and this is what happens when you give VA. So you bypass the first reactions of the CoQ pathway, and so especially CoQ6. Now this has been tried in non-renal models. It works. Other precursor works for CoQ7 and CoQ9. And basically, this is what I want to tell you. As a further reading, I would like to recommend this review that's written by Francesco Emma, Giovanni Montini, Samir Parik, and myself, that came out in 2016, but it's still quite updated. And last, I want to thank Billy Di Mauro, who was my mentor at Columbia University, Placido Navas, who was my mentor in coenzyme Q biosynthesis studies, my friend Fabien Pierrel, and this is what we usually do in Padova after we finish working and in non-epidemic times. And these are our collaborators, and this is my group, and most of the work that I've shown is done by these people. And I want also to mention Elisa Schiera, who wasn't in the picture at the time. I thank you for your attention, and I'm ready to take questions.
Well, thank you very much. Thank you so much, Leonardo, for this wonderful webinar. You just posted here on the last slide the upcoming webinars that all of you can read. Do you have Tanya? Can we bring up the questions for the polls?
Yes, sure. I will start the first question.
And I think, Leonardo, you can read them.
Wait.
Can you see them? Otherwise, I will do it.
No, wait.
Okay, I will see it. So mitochondrial DNA is transmitted only from the mother, from both parents, from the mother, but only to females, none of the above. So you can vote. There is an electrical discharge that will go on in your computer if you send the wrong answer. Okay. All right. I think, can you see the answers, Leonardo?
No, wait.
No. No, then I will read them to you. 83% got it right. It's only from the mother. I think we can go to the next question, Tanya. So the next question is, among mitochondrial disorders, steroid-resistant nephrotic syndrome is typically associated with complex 1 deficiency, coenzyme Q deficiency, all respiratory chain defects, mitochondrial DNA deletions. Please vote. And here again, I think most of you have sent the right answer is coenzyme Q deficiency.
Yes.
Is there another question? Yes. Is there another question?
Only when you
yes,
close the questions, you need to.
Okay, then I will let you comment and show the results. Okay. So I would read the questions for you and then you can comment the results.
Yeah.
Is there another question? So I will read it. Coenzyme Q is synthesized in the cytoplasm in the nucleus in mitochondria by a set of mitochondrial encoded enzyme in mitochondria by a set of nuclear DNA encoded enzyme. Okay, so I think you will have to make comments here. Can you see the answer, Leonardo?
No, yes, you wait. You need. No, I still. You have to close the. You have to close the poll. What?
So I will tell them. I will tell you the answers, and I think that this one deserves a comment from you.
Yes.
So 50% says answered in mitochondria by a set of mitochondrial DNA enzyme, encoded enzyme, and 41% said in mitochondria by a set of nuclear DNA encoded enzymes.
Yeah, this was a tricky question because the enzymes are in mitochondria. So the reaction takes place in mitochondria, but these enzymes are actually encoded by the nucleus and then transported into mitochondria. So this means that coenzyme Q deficiency is inherited as a Mendelian disorder, not as a mitochondrial maternally. It's not a maternally inherited disorder. So this is I probably wasn't clear enough in my when I explained it. So I put this question as just for me to see. It was control for me if I was clear enough. So this is important because all genes that control coenzyme Q biosynthesis these defects follow the standard rules of Mendelian genetics. So they're mostly autosomal recessive. We are discussing whether it's possible to have also some dominant phenotypes, but this is still a matter of debate. There are no X-linked disorders known to date, but we never know. But the majority is autosomal recessive. So this is important to provide counseling to the families, because an autosomal recessive defect means that if the patient is a children, the parents have 25% chance of having another patient, another child with the same disorder. This is important for them.
Very good. So Leonardo, we have a few questions for you.
Yes.
So the first question is from Simone Baldovino.
Yes.
Is there any differences between reduced or oxidized form of ubiquinol in treated patients with coenzyme Q10 diseases?
Okay. Now, there are very few data. In animal models, it appears that ubiquinol works better. But there are very few data on patients, because ubiquinol is also more expensive. So most people use ubiquinol, and it's made only by a few companies. We have had only one experience with ubiquinol, but it was not a pharmaceutical preparation, but it was marketed as a food supplement. And all our patients relapsed when we switched them to ubiquinol rather than ubiquinol. So my experience is negative. In the literature, in animal models, it appears that ubiquinol, so the reduced form, is better. But again, there are no studies yet in patients.
Okay, so Dr. Naveen Kumar Matewada is asking you actually 3 questions now, and because they're all related, I will just read them to you. When do you suspect mitochondrial cytopathies as a cause of steroid-resistant nephrotic syndrome? Do you routinely ask for inclusion of mitochondrial DNA in whole genome sequencing? And how do you monitor patients on CoQ treatment? So
Okay.
when do you suspect? Do you routinely sequence the mitochondrial DNA? And how do you monitor treatment?
The suspicion is usually is mandatory if you have a combination of renal and neurological symptoms. But we have seen many patients with what is it called, isolated renal symptoms. So it's something, especially if you have steroid resistant nephrotic syndrome, you must be sure that the diagnostic panel includes coenzyme Q biosynthesis genes. As for mitochondrial DNA, we usually you can do 2 things. You can basically look at the off-target reads. You can ask the lab. I am the director of my lab, so I do it myself. You can look at the mitochondrial genome on whole exome data. You can extract them, even if it's not specifically done. The problem is that it works well only if you have DNA from muscle. Otherwise, to be sure, we specifically, if you have a suspicion of mitochondrial disease, we sequence both the nuclear and the mitochondrial gene. I have to say that usually, if you have a mitochondrial DNA mutation, the renal symptoms are very rarely appear very rarely as a single manifestation. I have only that patient I showed, but you have to think. Probably in the future, there will be capturing novel capturing systems. We already do for panels that we can do both things at good coverage, but it's something to be in the future. And the last question was, how do you monitor patients? Basically, you see how do you
monitor patient on CoQ10 treatment?
Basically, you see how proteinuria goes. If you see a response, you see a drop in proteinuria. You may also want to add acetyl converting as what you call ACE inhibitors in non-acetyl angiotensin converting enzyme inhibitors to coenzyme Q if you have nephrotic symptoms. This association has been reported. But basically, the only thing now that it is not possible to do is you follow clinically. And usually, if you have a response, you see it. It's basic.
And Leonardo, we have, and I will close the questions here. So we have 2 more questions for you.
Yeah.
But if I may,
yes.
I think it's also important to insist that even though some of these patients have progressed to end-stage renal disease, I think one of the most important things is to continue to give them treatment to prevent their neurological deterioration.
Yeah, this is the point. This is the point. And in fact, there's a patient one of your patients that but you can comment on the patient that had developed the retinopathy. Remember, we once you told me.
Yeah, so, and again, we have one patient that had been treated. Unfortunately, he was diagnosed too late. He was already on dialysis, but we could prevent substantially neurological deterioration whilst his siblings actually did very poorly. So I think it's very important. I thought it was, I apologize for stepping in, but I just really wanted to make
this is an important point. No,
we have 2 more questions, and I think we need to go relatively quickly because our time is up. So Dr. Marina Akvan is asking you, should coenzyme Q10 treatment be a lifelong treatment? I think we just answered this, but I don't know if you want to comment.
It should be lifelong treatment. Yes.
And then we have the last question by Dr. Sadafa Sim. He is asking whether we should add coenzyme Q as an adjuvant to other immunosuppressive agents in steroid-resistant nephrotic syndrome patients.
Well, there is a paper from the Gasser group showing that PDSS2 variants were risk factors were found. And we have experience of finding heterozygous variants in patients that have other disorders. So it could be that it could be that they may be modulators. So it could be a good idea to do that. I think if you have patients, it would be a wonderful study to perform, and especially if you can correlate the presence of heterozygous variants in CoQ biosynthetic genes in patients with other deficiencies.
So this is a very good idea for a future study.
Yeah.
And if I may add again, I think that this treatment is actually expensive at the doses that you need to give it. So, but it's clearly, especially if you correlate it with some genetic variants, it may be a very interesting thing to do. So, Leonardo, this was a wonderful webinar. I think a lot of people learned a lot, and I thank you very much for your time. I checked, we had up to 55 computers that were connected. Usually, if you want to know how many people were listening, you need to double this number on average, because more than one person is listening behind each computer. And again, thank you so much. So let me
Thank you.
remind you all that the next webinar is on April 7. The speaker is Dave Silewski, and he will give us a lecture on neonatal nephrology. So thank you all. Thank you for joining in, and we'll hopefully see you and see you connected again on April. Thank you again,
Thank you,
Leonardo.
Thank you, Tanya. Thank you, everybody. Bye-bye.